Hello, myself Rajendra Shetty, Assistant Professor at Ganpat Parsikar College of Education, Harmal, Goa. Title of the unit, Biomolecules. Module name, Nucleic Acid, Structure of Nucleic Acid, Nitrogenous Bases, Nucleosides and Nucleotides. Outline, Nucleic acid and their importance, composition of nucleic acids, structure of nucleoside, structure of nucleotide, types of nucleic acids and their composition. Learning outcomes, learns the type and composition of nucleic acids, elucidates the structure of nucleoside and nucleotide, understands the importance of nucleic acids. Nucleic acids. They are present in the nucleus and cytoplasm of the cell. They control the important biosynthetic activities of the cell and carry hereditary information from generation to generation. There are two types of nucleic acids, that is DNA and RNA. DNA constitutes the genetic material in most of the organisms. RNA plays an important role in processes like translation and is also the genetic material of certain viruses. Composition of nucleic acids. Nucleic acids are biopolymers of monomers called nucleotides. Each nucleotide is made up of three important molecules, that is phosphate group or phosphoric acid, pentose sugar and nitrogenous bases. Types of pentose sugar present in nucleic acid. Pentose sugar present in nucleic acid are of two types, that is ribose sugar and deoxyribose sugar. As you can see in the structures, both the structures of ribose and deoxyribose are similar, just they differ at carbon number 2. In carbon number 2 of ribose sugar, there is presence of hydroxyl group, and in deoxyribose sugar, at carbon number 2, just hydrogen atoms are present. Ribose sugar is present in RNA and deoxyribose sugar is present in DNA. Types of nitrogenous bases present in nucleic acids. Nitrogenous bases present in nucleic acids are of two types, that is purines and pyrimidines. Purines are of two types, that is adenine and guanine. Adenine and guanine are two-ring structures. They contain 4 nitrogen atoms in them. At carbon number 6, on adenine, there is presence of amino group. On carbon number 6 of guanine, there is presence of keto or carbonyl group. Besides, there is also presence of amino group on carbon number 2 of guanine. Pyrimidines are of three types, that is cytosine, uracil and thymine. These are single ring structure with 2 nitrogen atoms in them. At carbon number 4 of cytosine, uracil or thymine contains carbonyl group, and on carbon number 4 of cytosine there is a amino group. At carbon number 2 there is a carbonyl or a keto group in all three of them. Besides, on thymine, on carbon number 5, there is a methyl group. As far as DNA and RNA is concerned, thymine is present in DNA and it is replaced by uracil in case of RNA. Other than that, the nitrogenous bases, that is adenine, guanine and cytosine, remains same in both the types of nucleic acids. Nucleoside. It is a component of a nucleotide. It is formed by bonding of pentose sugar with the nitrogenous base. The bond that is established is called as N-glucosyl linkage or N-glucosyl bond, or simply N-glycosidic linkage or N-glycosidic bond. Carbon 1 of pentose sugar is joined to 9 position of nitrogen of purine, which you can clearly see in the structure of the guanosine. Carbon 1 of pentose sugar is joined to first position of nitrogen of pyrimidine. This you can see in the structure of cytidine. Ribonucleoside is formed by bonding of ribose sugar with the nitrogenous base. Deoxyribonucleoside is formed by bonding of deoxyribose sugar with the nitrogenous base. Types of nucleosides. When the base connects or bonds with the ribose sugar, we get ribonucleoside. So, from adenine, we get formation of adenosine. From guanine, we get formation of guanosine. From cytosine, we get formation of cytidine. From uracil, we get formation of uridine. In the same way, when the bases they bond with deoxyribose sugar, we get formation of deoxyribonucleoside. From adenine, we get deoxyadenosine. From guanine, we get formation of deoxyguanosine. From cytosine, we get formation of deoxycytidine. From thymine, we get formation of thymidine. Here, we don't refer it as deoxythymidine because thymidine is only present in DNA and not in RNA. So, simply, instead of calling deoxythymidine, we can just refer it as thymidine. Nucleotide. Nucleotide is formed by bonding of nucleoside with the phosphate group. It is formed by phosphorylation of nucleoside. The phosphate group is linked to fifth carbon of pentose sugar by a phosphoester bond, which you can clearly see in the structures shown. Ribonucleotide is formed by bonding of ribonucleoside with the phosphate group. And deoxyribonucleotide is formed by bonding of deoxyribonucleoside with the phosphate group. Types of nucleotide. When ribonucleoside they join with the phosphate group, you get formation of ribonucleotide. So, from adenosine, when it bonds with the phosphate group, we get formation of adenosine monophosphate, or simply what we call as AMP. When guanosine bonds with phosphate group, we get formation of guanosine monophosphate, or GMP. When cytidine bonds with phosphate, we get formation of cytidine monophosphate, or CMP. When uridine bonds with phosphate, we get formation of uridine monophosphate, or UMP. The same way, when deoxyribonucleoside they bond with the phosphate group, it results in formation of deoxyribonucleotide. When deoxyadenosine bonds with phosphate, we get formation of deoxyadenosine 5' monophosphate. In the same way, when deoxyguanosine bonds with phosphate, we get formation of deoxyguanosine 5' monophosphate. From deoxycytidine, we get formation of deoxycytidine 5' monophosphate, and from thymidine, when it bonds with phosphate group, we get formation of thymidine 5' monophosphate. Types of nucleic acids. Nucleic acids are of two types, that is deoxyribonucleic acid and ribonucleic acid. We commonly call deoxyribonucleic as DNA and ribonucleic acid as RNA. In general, DNA is double-stranded and RNA is single-stranded. DNA is a biopolymer of deoxyribonucleotides and RNA is a biopolymer of ribonucleotides. Each deoxyribonucleotide is made up of three important molecules, that is phosphate, deoxyribose sugar and nitrogenous base. In the same way, ribonucleotides are made up of three important molecules, that is phosphate, ribose sugar and nitrogenous base. Nitrogenous bases present in DNA are of four types, that is adenine, guanine, cytosine and thymine. Nitrogenous bases present in RNA are adenine, guanine, cytosine, and uracil. Thus, we see that DNA and RNA, the difference lies in the pentose sugar which they carry, in which you see DNA has deoxyribose sugar and RNA has ribose sugar. Besides, there is a difference that is seen in case of the nitrogenous bases. The three nitrogenous bases, that is adenine, guanine and cytosine, remain same for both DNA as well as RNA. But in DNA there is presence of thymine, and in case of RNA there is presence of uracil. These are the references. Thank you.